To view animal cells, the source of the animal cells will be cheek cells. To obtain the cheek cells, we scrape the inside of our cheek. You can use a cotton wool bud, but sometimes that produces small strands of cotton wool or cellulose. So in this case, we'll use a wooden spatula. And we'll scrape the inside of the cheek using a wooden spatula. We now transfer the cells, saliva containing the cells, onto a microscope slide, and we just center it. To prevent the cells from dehydrating, we add a few drops of water. Normally about 3 drops of water would be sufficient. We now want to add a cover slip. The water could wet some of the lenses. So we add a cover slip and we place the end of the cover slip into the water there. And to eliminate air bubbles, we use a needle or something similar to lower the cover slip slowly, very slowly, and I withdraw the needle very slowly, and the water runs along the slide and pushes out and eliminates the air bubbles. We now transfer the slide to the microscope. Again, rotating the coarse focus so that this microscope stage is at its lowest point. Clicking in the low power lens, and we center it. So we bring the stage up as far as it will go. And then looking in, we focus. We may have to center the slide sometimes just to get the cells right. And we'll clip them into position. And focus. We can then see the cells at low power. To adjust the light, we can adjust the diaphragm, and that may give us a clearer image, or not, as the case might be. So we keep doing that till we find the image or the light intensity that suits best. To observe the slides then at medium power, we rotate in the medium power lens. Usually very little adjustment of the coarse focus here will do the job, and that brings it into focus. And finally, for high power, rotate in the high power lens, and this time using the fine focus, we can produce or see a clear image. To view stained animal cells, again we use cheek cells. Using the wooden spatula, we scrape the inside of the cheek to obtain the cells. We transfer the cells onto a microscope slide and again center them as before. This time, instead of water, we're going to use methylene blue. Methylene blue is the stain that's used to show animal cells. It highlights certain parts more clearly. Usually about 3 drops of methylene blue would be sufficient. Again, methylene blue would be damaging if it came in contact with the lenses. So to protect the lenses, we add a cover slip. Again, we touch the base of the cover slip down into the methylene blue and then gently lower the cover slip so that the air is pushed out and there are no air bubbles in the final slide. I'll just tidy up the slide a little bit. Transfer the slide onto the microscope stage, moving it down. Again, rotate in the low power lens. Looking down, bring the microscope the microscope stage up to its highest point, and always you're focusing downwards, so that means that the slide doesn't get broken or damaged by the lenses. If you were focusing upward, the slide might collide with the lens and crack and break, etc. So we're focusing in downward direction, and a little gentle focus, and the cells can be seen. We can then switch to medium power. Just place the clips in position first. We can switch to medium power and to high power and adjust or focus accordingly. All the time remembering to change the diaphragm and pick out the light intensity that's most suitable. The results of the unstained and stained cheek cells can be seen on the slide.